It's a real pleasure to welcome Sandy Knapp to present. Sandy is she's a prominent scientist, particularly renowned for her work in the Solanaceae that you'll hear about, I'm sure, the nightshade family that you'll hear about in today's work, but doing lots of still in the field work in the Amazon in Central and South America. She just got back from a fascinating trip to the Amazon. But she's also Director of Research at the Natural History Museum in London as well, and has had a range of books around botany and nature and plants, and is also a Fellow of the Royal Society and awarded an OBE for her services in natural history. So, over to you.
Thanks, Edvinder, and thank you everybody for coming. And so I kind of want to say, listening to all those other seminars you're going to hear, this is going to be something completely different. This is going to be something absolutely and totally and utterly different. And we were talking about, what kind of title to give this talk. And I mean, we can call it the Natural History of Nightshades, but it's also, I want to tell you about why taxonomy and why the kind of science that we do at places like the Natural History Museum is so important, not only for what it is that we do understanding the tree of life, but also how it links to all kinds of other science and becomes foundational, and that sort of verification of all of the things we do in nature recovery, all of the things we do in looking at distribution. If you don't have the taxonomy with good evidence, you actually are building on a castle on sand. So, oh, this is exciting. So this doesn't work. So I'll do it here. It's fine.
Yeah, just try now.
Okay. Yeah, so what I want to start out by saying is that science is a village. So all I'm going to talk about is work that's been done over many years in collaborations with many, many people, some of whom are here on the screen. Because you never do science as a single person. The best science is done when you're collaborating and communicating and talking with other people. And I found that one of the joys of doing science through my career. Now, we all know about nightshades, whether or not you kind of know about them through Harry Potter and mandrakes or deadly nightshades, but we all love members of the nightshade family. There are some of the potatoes are one of the 4 carbohydrates that provide, the 4 plants which provide 80% of the carbohydrates in the human diet, which makes them a really important plant, really important species. But there's lots of species of Solanaceae, as I'll tell you about. The family itself is a kind of middling diversity family. It's not big family like the orchids or like the daisy family, which have more than 30,000 species. It's kind of a middle of the road, pretty boring family. But actually, it's interesting and has always fascinated me because of the disparity and the imbalance in the number of species in the genera in the family. There's about 4,000 species in the whole family. There's 103 genera, but almost half of those species, well, a third to a half of those species are in one single genus. And I'll focus mostly on that, and the rest of it kind of tails off, and they're all really very lovely. Solanaceae inhabit a huge range of different habitats, from the deserts of South America to the high puna of the Andes, which is also an interesting dry habitat, which I'll talk about a little bit later, to the kind of intermountain ranges of Mexico and the southwestern US, to the savannas of Africa, to the central deserts of Australia, where they're hugely diverse. And I'll talk a bit about the central deserts of Australia later on. But the biggest generic level diversity and species level diversity of the family occurs on the eastern slopes of the Andes, which is where all that rain comes off the kind of Atlantic, goes over the Amazon, collects all kinds of water, and then dumps on the eastern slopes of the Andes, where you can get as much as 13, 14 meters of rain a year, which is a lot. It's very, very wet, and they love it there at that middle kind of elevation. So as I said earlier, what I'm really going to talk about is not the family, because I could do a thing of the kind of the magical mystery tour of the Solanaceae, which is also quite fun, because they also do very crazy things like are magical plants and are involved in witchcraft and all kinds of fun stuff. But I'm going to talk about the genus Solanum, which I've devoted my research career to understanding the genus Solanum. And this is kind of one thing I'm going to say now, and I'll say it again at the end, that if you're a student, never, ever let anybody tell you that something is too complicated to work on. I was told when I started out my career in taxonomy that I couldn't possibly work on this big genus because it was way too complicated. But they were wrong. So never listen to these people. One of the interesting things about diversity is that it's not evenly distributed. We know that plant diversity is not evenly distributed on the globe. There are areas across the tropics which have much higher diversity than the temperate zones. But also species diversity in genera is not evenly distributed. There are very few genera which have many, many species. Lots and lots of genera have very few species. And these big genera have always been termed monsters. And these are the areas where people fear to tread. But genera with more than 500 species, which are those things which young taxonomists are told, don't go there, it's too scary, there are too many species. Those are 25%, that's a quarter of all angiosperm diversity. And genera with more than 1,000 species are almost 14% of angiosperm diversity. So if we're going to understand angiosperm diversity, we need to understand these big, big genera. And Solanum is one of these big genera. These have been characterized generally as black holes, as that you just don't go there. They'll suck you in and take your entire career to understand them. And that's kind of true in a way, but they should not be considered as black holes, and these kinds of things shouldn't be considered as places where we don't go to do studies. So let's just start out by thinking about what a taxonomist like me does, and what is taxonomy? And I've showed this slide for years and years and years here at Oxford. I showed it in Kathy Willis's first MSc in Conservation and Biology a long, long time ago. And you can think of taxonomy as being three basic kinds of things. One of those, which we often think of taxonomy as being, is description, is describing new species or figuring out what the limits of species are. The other thing which taxonomists do is they look at evolutionary relationships, they look at phylogeny. But the other thing that we do is we look at identification, because without being able to identify all of these things, how are we going to be able to use them in the context of protected areas management, for example, or any of those things? So all of those things are part of taxonomy. Taxonomy has always been thought of as a solitary discipline, and a man, and it's always a man, sitting in the corner with his microscope, looking down his microscope and discovering great things and describing new species. And this is often in contrast to what people say is the paradigm for things like high-energy particle physics. This is the group of people at the University of Manchester who contributed to the Higgs boson, the discovery of the Higgs boson. So physics is often considered to be big science, partly because they have big equipment and work in big teams. They have big equipment like the Tevatron Collider at Fermilab. But if you think about what taxonomy has, it also has massive infrastructure, which is museums. And museums have been built up over hundreds of years rather than tens of years like the Fermilab. So we have big infrastructure as well. One of the things we might not be great at is working as teams. So that's what I kind of want to talk about. But we also have a super advantage over physicists, is we have the field. We have the place where organisms interact with their environment and do what it is that make our planet as vibrant as it is. So Solanum has always been, Solanums are pretty much all the same. They all have a kind of basic floral ground plan. They kind of look more or less the same. If you squint, all of those look pretty much the same. So squint. But they've been divided up into lots and lots of different genera. Solanum in the past was divided into lots of different genera, largely based on flower morphology. There's tiny little differences in flower morphology. When we first started to do DNA sequencing, however, we discovered that in fact, all of that diversity was actually monophyletic with really high support. And this is 20 years ago. This is very early stage DNA sequencing. And Solanum can essentially be divided into two big chunks, one of which is monophyletic, meaning they're all each other's closest relative, which are the spiny Solanums, and they have these prickles. They're actually technically prickles. They're outgrowths of the epidermis of the plant. So there's spiny Solanums, and then there's the non-spiny Solanums, which are paraphyletic. So they're divided into these two big chunks. And those big chunks and those basic clades have remained the same since 2005. No matter how many genes we throw at it, no matter how much we do, it's basically the same. So we've done these iterative phylogenies, looking at Solanum, looking at whether or not it's using more genes to look at monophyly, and it's always monophyletic, and the clades are always basically the same. And I'll talk a bit about that later. But not only does Solanum have the highest species diversity in the family Solanaceae, it also has the highest lineage diversity. So this is a phylogenetic tree of the whole family, and you can see that the number of lineages within Solanum is very, very high. So it's a place where there's a lot of evolution going on and a lot of differentiation. Solanums come in all kinds of body shapes, and many people don't think of plants as having bodies, but plants have bodies just like animals do. They're just different to ours. They come in very small. This is a tiny little plant from the high Puna of Argentina, which is doing flowering, fruiting, and a bud, and that's my thumb holding it. And they also come as large, as huge trees, and even as forest trees in the Amazon. So you get this incredible variation of body shape across this very, very large genus of more than 1,200 species. One of the advantages of working on something like Solanum is that people are really interested in it because we eat them. And so they have a use to us. I mean, all plants, of course, have a use to us. But one of the things that happened to the group of people that I work with is that about 20 years ago, the National Science Foundation decided to invest in a global monographs, a global treatment of the entire genus Solanum. But what they said at the time is, we'll give you this money, but one of the things you must do to get the money is to interact with the genomics community. Now, the genomics community, now everybody does genomics. We all use genomics and phylogenomics as a way of looking at the world. But 20 years ago, that was not the case. And the people who were doing genomics in Solanaceae were the people who were looking at plant breeding, looking at molecular breeding, molecular ways of improving the crops that we so much depend on. So this is the Tomato Genome Consortium. So this is in 2004. So this is more than 20 years ago. I'd like you to notice the gender balance of this slide. I took the picture, so you know. But so these were the people who were looking at the tomato genome. And now we do genomes a dime a dozen. And in those days, we were doing them bacterial artificial chromosome by bacterial artificial chromosome. So it was very, very different. So this was me, a taxonomist, needing to interact with all these people over, in order to get the money to do the taxonomy of Solanum. We ran into a bit of a problem towards the beginning because we discovered, as part of doing phylogenetics, is that potatoes and tomatoes are each other's closest relatives. But the problem was that tomatoes had always been called the genus, had since 1768 been called the genus Lycopersicon. And the entire tomato breeding industry was based around this genus name, Lycopersicon. So this was a bit of a problem. And one of the things that we did to try to kind of bring these communities together was say, one of the things about names is that applying names is not something that's set in stone. Each name is a hypothesis. And the hypothesis is where you cut that tree. Where do you cut the tree of Solanum? Do you cut it here and we have a monophyletic Solanum? Or do we cut it on the red line and we have loads and loads of different genera, all of which you can't tell apart and whose relationships are unclear? So there was a big, I remember going to the Plant and Animal Genome meeting in 2004 in San Diego, which is quite scary. It's thousands and thousands of people doing genomics. And I remember sitting in the audience. I'd been invited to give a talk. I remember sitting in the audience and writing down in my little notebook an entire list of words that I didn't know what they meant. I had no idea what they meant. And I thought, okay, I'm going to stand up and give a talk about phylogenetics, and these are going to be lots of words that you guys don't know. But I got a very hard time from these people for changing the name of the genus Lycopersicon because it was part of a monophyletic Solanum. They gave me a really hard time. They said, you can't do that, you're going to ruin the entire tomato breeding industry. And I said, well, there's three possibilities for this. And the person who did the phylogenetics to really determine the relationships of tomatoes and potatoes is Iris Peralta, who's from Argentina. She's from Mendoza in Argentina. So I said, there's three things you can do. You can either ignore us completely, which is fine, I don't care. That's not my problem, it's your problem. Or you can change the names of the potato to Lycopersicon tuberosum, because remember, potatoes and tomatoes are each other's closest relatives. And you can change the name of the eggplant to Androceros, which is fine. Or what you could do is what we've done is to change the name of the tomato back to Solanum lycopersicum, which is how Linnaeus originally described it. And they kind of thought about this for a while. And basically, due to kind of this close collaboration with plant breeders and talking to them and learning this vocabulary, learning how to talk to a community which is completely different to the one that we came from. And this is Danny Zamir and Zach Lippman, and you'll hear about Zach Lippman a bit later on, is that the tomato genome was published in Nature as Solanum lycopersicum. So we completely convinced this community to take taxonomy and systematics and phylogeny seriously. So I'm going to switch gears again now and talk more about what the kind of things that we do as taxonomists and how this coming together of communities has both strengthened the work that we do, but also strengthened the work of our collaborators as well. Solanum has always been one of these big genera. It was one of Linnaeus's largest genera, and it really exploded in species diversity as soon as Alexander von Humboldt came back from his collecting expeditions in South America, which brought back all of that diversity from South America, all of that diversity from the Andes. Because if you remember where Humboldt went, is he went from Venezuela down the Andes, down the back of the Andes, which is where all these things are so diverse. So it's always been a big genus, so it's always been a bit of a problem, and people don't really like to go there. One of the other problems with working as a taxonomist in Solanum is that we have a problem with something called synonymy. And you guys all probably know what synonymy is. Synonymy is often considered to be a mistake, that taxonomists of the past have made mistakes because they've described something as different when in fact it's not. But actually all synonymy is the accumulation of evidence. Because if you think about collecting in Africa and you had a plant from Senegal and a plant from Congo, and they looked a little bit different. One had white flowers with pink spots and one had pink flowers with white spots. And they look quite different if you have two of them, just two specimens, just two instances. Roll on 100 years and there's 200 specimens in between, which show all of that variation in between. One of those names becomes a synonym. And that's not a mistake. It's about evaluation of evidence, which is why species names are all hypotheses. So synonymy is a big problem in Solanum because of these cultivated plants, because the taxonomists of the past would often name cultivars with botanical names. The cultivated potato itself, this is a paper we did with some scientists from St. Petersburg in the Vavilov Institute. The potato itself has 600 synonyms. So working out what all those are and how it all works takes a long time. So that's a bit of a problem. So we're drowning in synonyms. The average synonymy for flowering plants is 30%. In Solanum, we have 80% synonymy, which means we really need to do a lot of work sorting out the names of the past. But there's also a lot of sorting out the names of the present and looking at the diversity of the present. And that can be quite exciting. You can go on very fun roads in places like Andean Peru. You can find really lovely things that don't have names. This is a plant my colleague Gloria Barbosa, again from Argentina, and I described a few years ago. Actually, we described it during the pandemic because I was in Argentina until 2 days before the entire UK shut down. I was in Argentina, I came back, and then the whole place shut down. So thank goodness I got back. It would have been fun to be in Argentina, actually. But you can describe these new taxa, which is exciting, but actually almost as exciting as describing new taxa is figuring out the limits of taxa, figuring out what all the names are and what you actually have in front of you. What are these units that you can work with? But this all depends completely on collaboration. And it's really important when you work in a group like Solanum, whose major diversity is not in places like Britain or any of the Northern Hemisphere, that this is all done in collaboration with local botanists. And this young man is Paul Gonzalez, who was an undergraduate student on one of our very big trips in Peru in 2010, 2012. And he's now completed his PhD, and he has done the most beautiful revision of violets of the Andes that I've ever seen. It's absolutely gorgeous. So it's really great to keep these relationships going and not, it's so often what happens with taxonomists is they go somewhere, collect things and take them away. And that's the old paradigm. So I have a rule of thumb with the people who work with me that if they go into the field, to collect, they have to spend at least half the amount of time that they're there in the field, in the collection, in that country, working with the local scientists. Because that's the only way that those collections get annotated and updated identifications. So that's really important. So these are some of the things we did as part of this big project. We had a website, we did lots of monographs, we monographed, we did the taxonomy of lots and lots of these clades. The green ones are ones we've done. The purple ones are ones that are still left to do, but actually there's a Peruvian student who's done this one, whose manuscript we're just editing. So this is all getting done. It's amazing. Took longer than the 4 years we had for the grant, naturally, but we're doing it. And then we also provided these online keys. So one of the problems with these big genera is it's really hard to get into them. It's really hard to figure out where do you start if there's 1,253 species. So figuring out how to make those keys has been really important. One of the other important things that has happened from working together as a big group is we've gone to many herbaria in countries where these species are very diverse. And this is my colleague Leandro in the herbarium at IMPA in Manaus just a couple of months ago. And you can map the world in solanums. All those dots are solanums. So basically solanums are everywhere. So they're marvelous. And we have 72% of these we've georeferenced. And this is important because then we can use these data. So it's not collecting data for collecting data's sake is a bit boring, because then what do you do with it afterwards, which is what's interesting. And having all those data mean that you can actually look at things like species richness and net diversification. This is the work of a PhD student, a Colombian PhD student, Susi Echeverria Londoño. She did a while ago now. And what we first started looking at species richness, and we knew that the species richness of Solanum was the highest in the Andes and in southeastern Brazil. We've known that for a really long time. That's clear. Like I said in the beginning, that the biggest diversity is on those eastern slopes of the Andes. So we kind of expected diversification to also be very high in those areas of high species richness. That makes sense, doesn't it? Where you have a lot of species, there's a lot of diversification. It turned out completely, against all odds, it turned out that that was not true. That the highest diversification rates are in Africa and Australia, which are in one clade, which is the spiny solanums, those spiny solanums. And that was a complete surprise. We didn't think that was going to be, we thought it was a mistake, so we ran all of the things again, and it wasn't. So we have this interesting pattern of species richness and net diversification being the mirror images of each other. And the other thing is this came on together with another surprising result, was that all of the spiny solanums that occur outside of the Americas are each other's closest relative. For many years, it was thought that all of these spiny solanums that were outside of the Americas were members of groups that had originated in the Americas and perhaps gone out of the Americas. But nope, they're all each other's closest relatives. So there's been a really interesting biogeographic split there. And so we think, based on Susie's work, that the sister group of these out of the Americas, which now have a name which is called Hemigeacanthos. We call them the Old World, but I'm trying to get away from calling things Old and New World because I think that's a value judgment. But the sister group of these is a tiny little group of species which occur in the southwestern US and in Argentina. So that's often the case. You often get these very small groups being the sister group of really big groups, because the out of the Americas species is 400 species. It's huge. So we think this has to do with the fact that these plants, which went outside of the Americas, were pre-adapted to these drying periods, because dating this tree, you can see that I'll go back, is that all of these are in kind of drier areas, like the center of Australia. And if you look at the distribution of different types of underground organs within the genus Solanum, you have things that are non-geophytes, so that's everything that kind of is just a normal plant. And then you have the potatoes. So there's the geophytes. Here are the potatoes, which are cold and dry. And these are the rhizomatous species. These are things with underground rhizomes, which allow them to spread. And those are very, very diverse in these dry areas and are hot and dry when you model their environment. So that means that you essentially have what is a controlled experiment in Australia. Is the highest diversification rate that Susie found was in these species that were outside of the Americas. But interestingly enough, in Australia, there are two clades which are completely endemic to Australia, one of which is much earlier than the other. And that one is confined to the wet southern part of Australia. So we think that during the Miocene drying of the center of Australia, these rhizomatous species, because all the species in Australia are rhizomatous, really exploded in diversity when all these arid habitats were opening up and they were empty. So we're now working on the Australian solanums to look at that. So these plants that went out of the Americas went to the right place at the right time with the right traits to be able to explosively diversify. And the Australian solanums are really interesting, partly because of the 179 endemic Australian solanums, almost 100 of them were described since the Flora of Australia was written. So we know a lot more now, and there are lots more still to describe. So a lot of this depends upon phylogeny. A lot of these hypotheses of how things happened and dating phylogenies depends on having a really good phylogeny. And as I said earlier, you know, that early phylogeny, we still have basically the same topology as we had when we were using 3 chloroplast regions. We basically have, this is the latest, well, this is the penultimate phylogeny of Solanum. We haven't published the new one yet, but this one was with 742 of the species, so that's more than half the species of Solanum, which kind of, everybody says, well, why don't you have them all? And it's because there's kind of a lot of them. And we use 9 genomic regions for this, but the clades are all perfectly stable. They're basically completely stable. We have good groups, but the relationships between those groups are very unclear. Remember that diagram where I was cutting the tree and things were all really, you didn't know who was related to who. It was like a comb. We still don't, the relationships between the groups are still not particularly clear. So one of the things we did with this is to look at areas of discordance. Discordance in phylogenies tells you where there's interesting biology going on. And there are 3 areas of really big discordance in Solanum, one of which is right at the base of Solanum, and the other of which is at the base of the spiny Solanums, remember that really big clade, and the other of which is at the base of those Solanums which are outside of the Americas. So those are areas which either there's been hybridization events, there's been a lot of incomplete lineage sorting, or whether evolution has happened too fast for us to be able to resolve it. So distinguishing amongst those things can be quite difficult, and it requires more genes and more species. So that's what we're partly working on. But there's also, we've had discordance in particular areas of our phylogenetic tree for a long time, and one of those has to do with the potatoes. So this is this woman selling all kinds of lovely kinds of potatoes in Peru in a high elevation market. And potatoes are part of an area of the Solanum tree which is highly discordant. There's 3 clades, one of which is the potatoes, one of which is the tomatoes, remember they're each other's closest relative, and another one is a very tiny little clade called E. tuberosum, which occurs in Chile and on the Juan Fernandez Islands. And those have always flipped and flopped. We've never known who's each other's closest relative. So together with some of these genomic plant breeding colleagues, so meanwhile, behind all of this fun kind of Australian stuff is there's genomic conversations happening with plant breeders. We decided to look at discordance at this potato-tomato E. tuberosum node. Because Sangwang Wang, who did this work with us, is very interested in developing a diploid potato, and we can talk about diploid potatoes and why diploid potatoes are important later. But what we found is that there's high discordance at this area, and this happens around the mid-Miocene. Remember, that time of the mid-Miocene seems to be a really key time in evolution of this particular group. And what Sangwang and his colleagues did is based on our sampling and our identifications of all of these wild species of potatoes, is they did genome sequencing of all of these and found that at particular areas in the genome, there were 2 genes which govern the formation of tubers. And those 2 genes, which come together to make tubers, that govern the development of tubers, half of them were derived from E. tuberosum lineage, and half of them were derived from the tomato lineage, which means that sometime in the past, and we estimate this about the mid-Miocene, there was a hybridization event which brought the ancestors of these two lineages together to create something which later went on to become the potatoes. And this actually jives really well with ooh, that's quite fun up there. It goes quite well with some of the things that Susie found. That green blob is the potatoes. And that green blob, it means that the potatoes are diversifying faster than the background rate in the genus. And so we think that the reason that this is happening is that this hybridization event kickstarted something that was happening concurrently with the uplift of the Andes and the creation of those colder, drier habitats which favor those storage tubers, and that the Andes and their uplift actually contributed to the diversification of potatoes, which are completely endemic to the Andes. This needs testing with better data and more refined methods, but it's a really good hypothesis to go on. And it wouldn't have happened without the phylogeny, the taxonomy, and the genomics, people all talking to one another and attempting to understand each other. So one of the great things about having a phylogeny that you think, okay, this is great, is you can map characters onto it. So one of the things we've also done is mapped characters onto the phylogeny. And some characters flip and flop all over the phylogeny. And this is one of them. There's 58 gains of red fruit across Solanum. So red fruits might not all be homologous. Red, the chemical that causes red, comes in lots of different varieties. Lots of different chemicals cause red color. But the fact that this isn't a good character for defining groups is actually, that's why we did this. One of these characters, though, piqued the interest of another set of people, and this is Zach Lippmann, who I showed you in the picture earlier. There's a single gain of prickles in Solanum. Remember, there was a big monophyletic group, which was the spiny Solanums, the prickly Solanums. A single gain, but within that clade, there are 14 losses of prickles. So Zach got really interested in what was going on with the loss of these prickles, what was governing the loss of the prickles. So they did, we constructed a sampling across the whole of the genus, picking sister taxa, which had prickles or didn't have prickles, and then polymorphic species, which had prickles in some individuals and not prickles in others. And there are some species which have prickles on one branch and no prickles on another, but that was kind of out of the picture. And so these red ones are the prickles that are lost or suppressed. And what they found doing whole genome sequencing is that the loss of prickles was due to mutations in this thing called a log gene, which stands for lonely guy. Who knows how genes get named? I mean, I get how species get named, but genes, I don't know. And it's a cytokinin hormone regulation gene. And what's really interesting about this is that something that seems as though it's homologous, which we, as taxonomists, we're really interested in homologous characters. We're interested in things that are the same for the same reason. So something that looks homologous actually occurs in lots and lots and lots of different ways. So these are all teeny little frame shift insertions or deletions, and even sister taxa have it different. So again, working with Zach's lab, we've done a pangenome of all of Solanum, which means we have a genome of all of the clades of Solanum. And again, something that looks as though it's the same, which is having multilocular fruits. You know how when you get beefsteak tomatoes, they've got like lots of compartments in the fruit. That happens a lot in cultivated plants. So people have always assumed that the multilocular fruit of this species, which is called Solanum ethiopicum or gilo, and the tomato is due to the same gene, which is called it's called F2. It doesn't really matter what it's called. But it turns out they're due to completely different genes, completely different gene pathways. So something that looks as though it's homologous actually turns out not to be, which I think is, as a taxonomist, that's kind of scary, because you think I'm going on these characters and I think these are caused by the same reason. But actually it's really exciting, because it means biology is more interesting than we thought. So that's kind of a journey of a taxonomist and one of the things that I think is important about working on something that's big and complicated and that people have told you not to work on is that it leads you down pathways that you never ever thought you would go down ever in a million years. And it leads you to talk to people who are not like you and who don't think like you. and don't think the same things are important. But yet, if you actually talk to one another, it makes a big difference. So black holes are not so bad after all, because they turn challenges into opportunities. So one of the things we laid out at the very beginning of this Solanum project is what the challenges were. And the challenges are time, access, and institutional commitment. Well, time is always a challenge. That's never going to go away. Access to information is also a challenge. It's less of a challenge now than it used to be. Commitment of institutions is also always a challenge. But actually, those challenges that we thought were problems at the beginning of our big project turned into huge opportunities. One of those is working as a collective. We now have a collective of Solanaceae taxonomists around the world, all of whom are working together. The most recent paper we've published is the new phylogeny of Solanum with a new classification, and it's got lots and lots of authors from about 30 countries. Credit for effort, we thought was a problem. People were worried about contributing to a big collective project because they wouldn't get credit for the effort. But that turns out that if you start working collaboratively, that turns out not to be a problem. Common goals are easy to set if you're willing to think in somebody else's shoes. And specimens are a problem, because specimens are a problem. Because Linnaeus had 2 specimens of the cultivated tomato to look at when he decided what it was called. Whereas when we did a revision of the cultivated tomatoes in 2008, we had thousands and thousands of specimens. And to make a decision about each one of those actually takes time. And that's one of the ways in which AI may be able to help us. But I'm not sure I would ever really trust it. And mission creep, which is always thought to be a problem in projects, actually can mean better reach. It can mean that you connect to more people and learn new things. So I think it's important now to think about these things, to think about how we construct our projects, because we are confronted with that perfect storm of environmental change right now. Because we have a lot of anthropogenic effects, not only from climate change, but also from land use change through human intervention. So it's a time that we need to think about working together much more than we ever have before. And I think collaboration is more complicated than we ever think it is. Because we often think about it that we pull different teams together, we have a common goal, we kind of exchange knowledge, all of that's really good. And that's the bit of the iceberg of collaboration that's above the surface. But this amazing diagram from the Turing Way, if anybody hasn't ever seen this or used it, it's an absolutely fantastic resource, is all of these things that are below the surface are really important for making collaborations work. And kindness and being explicitly open, building those diverse teams and being inclusive are super important for making collaborations work well. And so I just want to kind of end by giving you one of my favorite quotes from Alexander von Humboldt, who is one of my great heroes of biology. And he said the most dangerous worldview is the worldview of those who have not seen the world. And you could pretend that meant that people who didn't travel had a problem. But actually, what he really meant is that it's important to see the world through someone else's eyes and to see the world as someone else sees it, because that's the only way in which your worldview will be amplified and increased. So I think he was very wise, Alexander von Humboldt, even though he's a long time ago. So I've had funding for all of this work from a ton of different people. National Geographic funded lots of our fieldwork. The National Science Foundation was brave enough to have an entire program which was about doing monographs of complicated big things. NERC has funded us, BBSRC, and our own institutions have been absolutely great, the EU as well. And if you ever want to find out about solanums, this is our website. This is our new, shiny new website, which is hosted by GBIF. And we're just putting it together. So once it's there, I want people to kind of comment on it and say what's wrong with it, because that's the only way anything gets better. And I'll just leave you with those amazing people that I've worked with for so many years. And just to remind you that science is a village, and the way to get it done the best way is through collaboration and seeing the world through other people's eyes. So thanks very much.
Okay, thank you, Sandy. Fascinating. And as you said, a good introduction to the power of taxonomy giving insight into the world. Okay, we can open up to questions. Anybody want to kick off a question?
Amazing. Thank you so much. This was so interesting, and I loved just all of the discussion about collaboration. I was wondering if there is a species of Solanums that you have yet to see out in the field that is kind of like your white whale,
Gazillions.
or gazillions.
Gazillions.
Do you have like a top two that are kind of like up at the top?
I really want to see some of these Australian ones because they're wacko different and they're really interesting. And a lot of the times when I had this National Geographic funding, I would not go in the field. I would send the students and the postdocs in the field because I spent a lot of my career in the field, you know, collecting and doing stuff. I used to work for Missouri Botanical Garden as their kind of man in Panama. I was their man in Panama. And I was given a truck and a thing to drive around in and a horrible hovel to live in, and just collected plants for a whole year, which was really great for understanding plants in general. But I'd love to see some of the Malagasy. There's a Malagasy Solanum, which is called Solanum mahoriense, which you know Chinese lanterns that have the orange kind of inflated calyx. Well, this has got an inflated calyx that's covered with spines. It's just the coolest looking thing. I would really like to see it. But no, there's a lot. I could go on and on and on. But I saw one of my great Solanaceae wonders of the world that I've always wanted to see, which is a tropical forest tree called Duchiodendron, which is endemic to the area around Manaus and in the kind of in the sort of Rio Negro sort of basin. And it's a huge forest tree, 30 meters tall. And I've always wanted to see it, and I finally got to see it when I went to Manaus in November. And it's like a tree that's like not doing a very good job of being a tree. It's got kind of really pathetic buttresses, and it's got sort of really soft wood, and it's like it's stuck somewhere where it really oughtn't to be. It's just, and it's got those fruits which were obviously eaten by megafauna that now just rot on the ground. So so seeing it was exciting. There's one in the way back as well.
Thank you. Thank you for the. Oh yeah, you can hear me. All right,
Yeah.
good. Oh, perfect. I won't do that. Thank you for the talk. Fascinating. I wondered. You spoke. You touched on the tubers and sort of the different root morphology being probably adaptations to sort of facilitate this expansion of the genus into maybe, I mean, out of basically the Andes Mountain, out of the Americas and into Africa and Australia. But it also seemed like one of the shared traits, functional traits, were the spines, they weren't spines, they were
Prickles, but
prickles,
we call
yeah.
them spines because
Exactly.
spiny solanum sounds so much better than prickly solanum,
Yes.
doesn't it?
But I just wondered, so have you done any research or sort of any consideration as to what is the point? What was the adaptive advantage of having those prickles as these species moved into these drying Miocene habitats?
Well, there are prickly solanums everywhere. I mean, the thing is, there are prickly solanums everywhere. And so that subgenus, that group called Leptostomonum, the spiny solanums, occur in all habitats. They're not just confined to these dry areas. And people did, in the 1970s, people tried to figure out why there were spines on solanum. So they did all these experiments with caterpillars, and they put caterpillars on, and caterpillars would die, and then caterpillars wouldn't die, and they hypothesized that the prickles would keep animals from eating these plants. None of these things is true, because if there's an absolutely fantastic YouTube video of impalas eating the fruits of one of these spiny solanums, and they just kind of grab them off the plants, and they don't seem to care at all whether there's spines. So nobody's ever really figured out what they're for, and they might not be for anything, which is partly why, I mean, if they're not for anything, and this loss of spines is a stochastic process, then you would expect what we found with the lonely guy genes is that the loss is not all due to the same thing. It's all due to all kinds of little tiny random mutations and frame shifts. So it's not necessarily phylogenetically important. It might be one of those things that you have because you have it, like tailbones in human beings. Is that it's I guess one of the things that's the hardest sometimes is we as people want everything to have a purpose, and I think spines may have a purpose in some areas, but as a character on the tree, as a single evolution of that characteristic, the single acquisition of that, we might not know, we might not ever know. But it's a really good question, because I keep wanting to go back to those 1970s papers, because they did some absolutely hilarious experiments. They were great. There's another one there.
I can see that they originated around 30 million years ago. So what was the mechanism for dispersal across the continents?
So we've just redated. The other thing we've done, which I didn't talk about, because we do all kinds of different things. So one of the other things that we've done is and this is a postdoc, a very talented Argentinian postdoc who's now a professor at the University of Finland in Helsinki, at the Helsinki Museum. She is very interested in paleobotany. And so what she did during the pandemic, she got all these fossil seeds. So the fossil record for Solanaceae is not great. So we did that tree that I showed near the beginning was a dated tree that said that the whole family was about 20 million years old, blah, blah, blah. But there are these amazing fossils which have come out of Territorio Chubut in Argentina, which are beautiful inflated calyces, which are 53 million years old. So they're Eocene. And they look like Chinese lanterns. And so there's been this debate about dating, and dating using molecular data is always a bit dodgy. Everything's always too young. So what's happened throughout the history of trying to date phylogenetic trees of plants is they're always too young. The first ones are always too young, and they progressively get older as we find more and more fossils. Well, what Rocio did is she took all of these fossil seeds, and we scored hundreds and hundreds of extant species of Solanum for the characters that we could see in these fossils. And then we did this whole reconstruction of dating. So there's a new paper which is coming out, I don't know, maybe in a few months, with a new dated tree. And it looks like the whole family is pre-Eocene, is Miocene. But that all of those, the pre-Eocene, so 53 to 60, 98 million years, it's a Cretaceous. So it's really old. But all of these, the datings of these tips, where we have these things happening in the Miocene, none of those have changed based on this data. And so this is kind of what happens across angiosperms when you start to do dating, is you find that the basal lineages, the early branching becomes older, but the tips actually don't change that much. A little bit, they become a little bit older, but they don't change that much. So this redating will be really interesting to try to, so we included all of these fossils. And as part of this, we've ended up describing about 10 new seed fossil genera of Solanaceae as well. I mean, these seeds, I still have on my desk, I should probably not admit this, I still have on my desk at the Natural History Museum the entire Solanaceae fossil seed collection from St. Petersburg, because I can't send it back. Because the guy from St. Petersburg said, don't send it, don't send it, don't send it, because this is a problem. We have a problem in Russia now. So I'm keeping it. Safe.
You can edit that for the recording if you want.
Yeah. No. This is an agreement between us that we do not want this really important collection to be lost. So it needs to be hand carried back to St. Petersburg when we can do that.
And this dispersal.
I think because the family is so old that a lot of this dispersal happened across the Antarctic.
Okay.
Across Gondwana, because you look at some of these lineages, and I think understanding the Australian lineages better will help us with that, because in the Cretaceous the continents were apart, but they weren't that far apart. They weren't that far apart, and a lot of these Australian solanums now have dry fruits. There's one that a colleague of mine described from the Northern Territories called Solanum ossicruentum, because it's really hard. It's like a hard fruit. I mean, they're like rocks, and you cut it open and it has red sap, so it bleeds. So it's kind of this bizarre kind of hard thing that bleeds. And so not all of them are birds. So we can't, I don't know, but those could be subsequent things that happened in Australia. So we need to understand those Australian solanums better and the characters better within those.
Okay, great.
Yeah, there was a question back there and there.
Thank you so much. That was super interesting. I think everyone in this room probably has a really strong kind of understanding and appreciation for taxonomy and the study of evolution.
Well, you guys are exceptions.
And I was just wondering, in a kind of in an arena increasingly kind of dominated by the climate crisis and biodiversity crisis. I noticed you had a really amazing kind of array of funding and kind of resources. Is it becoming harder to persuade kind of funders that taxonomy is really valuable kind of thing to be doing? And how is that changing at the moment?
It's interesting because taxonomy for its own sake used to be a thing that people did. But actually science in its own sake is something that none of us do. We all do science as a way to ask and answer new questions. And I think if you frame taxonomy as an end in itself, nobody's going to fund that. Whereas if you talk about taxonomy as being part of a solution to something, to a problem, it's about having a problem. So I did a fellowship. I did a bunch of fellowship interviews yesterday, and I asked one of the candidates. I said, "What's the problem you're trying to solve?" So it's not so much what's the technique you're going to use, but what's the problem you're trying to solve. So if the problem we're trying to solve is why is it do we have potatoes, then taxonomy was a really integral part of that, because we needed to know what all those wild species of potatoes were. We needed to know how they were related to one another. We needed to know what the sister group of that was, and we needed to know, and that's where the collaboration thing gets built in. And that's why sometimes it can be difficult because none of us have a lot of time. Time is that thing that is always in short supply. But devoting the time to actually talking to people about why it is that it would be good if we did this together. Let's do this together. Let's try and think about this as a joint problem is really important. It's like going into local communities. It's like going into local communities and working with local communities. Is you don't just go into a local community and ask them lots of questions about their varieties of cassava or whatever it is they're growing. You spend time and ask them about their children and about their families and about their crops and about their this. And that's, I can remember working on a project like that when I was just after doing my PhD and when I lived in Peru. And the person who I was doing the project with was impatient about the amount of time that we spent in the communities, but the time that we spent was worth double its time, because we developed relationships and were able to really think about that through the people who were growing those plants' eyes. So I think it's about solutions, really. There's one back there.
Thank you so much for the talk. It's always fascinating to see people that are really passionate about the subject. And me as an entomologist, I get excited when I see people that do. I can really sense the joy of your work. And I guess I have two very basic questions that are, the first one is, so I get that this group is very diverse because it's really old and because of the Andes, and they had the right genetic conditions to diversify. But I wonder to what extent have the chemical composition, like the alkaloids and the lack of herbivory, has helped to the diversification of the group? I guess that would be my first question. And the second one is, I realize that these are maybe two very different things, and I realize that there are like these tensions between taxonomists, classical taxonomists, and maybe people working on barcoding, for example. But I wonder to what extent could a tool like that help for the synonyms, for example? But because I know that for entomology that has helped with the classification, but I don't know if this applies for this case or not.
Barcodes are easy peasy in insects and fungi because you can do CO1 barcodes. You can use one little region and you've got a barcode. Plants are super difficult. Plants have really complicated genomes. And so we have no proper barcoding in plants that really work at all. And if you think about barcoding, so barcodes are a character, right? And what taxonomy is, it's about comparison. All taxonomy is comparative biology. There's nothing special about taxonomy. It's just comparative biology. And so if you take those comparisons and you use a single character, that would be crazy, wouldn't it?
Yeah.
So it would be crazy to just use barcodes, because barcodes are a single character. So it's about bringing things together, and barcodes are a really, for insects and for fungi, barcodes are a really important characteristic. They may not tell us species limits, but they help us with identification, particularly in fungi, where you have anamorphs and telomorphs, the sexual and asexual stages of fungi, which look very, very different. Barcodes have been revolutionary in enabling pulling together anamorphs and telomorphs. So I guess, so that's the kind of second question, and now I've forgotten the first one again.
About the
Oh, about the herbivory.
yeah.
So there's a group in the Andes, there's a group of butterflies called the Ethomyine butterflies who are specialists on Solanaceae. And with a PhD student of my ex-husband's, we did a paper looking at the diversification of that group of butterflies in conjunction with the rise of the Andes and the phylogeny of them and the phylogeny of Solanum. And they don't really match together very well at all. And but there are these specialist groups of butterflies, and they are very undiverse compared to the genus Solanum. The genus Solanum is much more diverse. And there are none of those butterflies in Australia, for example. So they probably aren't a driving force. The diversity of Solanaceae is likely to be a driving force in the diversification of the butterflies rather than the other way around. I mean, that's just that's of course what I would think, isn't it?
Thank you so much.
There's another question over there. And then there's one here.
Buzz pollination. This is a feature of the one Solanum species I know from temperate Europe.
It's a feature of all Solanum.
Okay, very good. Yeah, I wanted
Every single one is
Excellent.
buzz pollinated.
And what are the sort of the implications then for sort of related to that question? Bees sort of discovering how to use this. I mean, the co-evolution of I guess bees, which are the main buzz pollinators, and then Solanum.
Yeah, so buzz pollination, so buzz pollination is an amazing pollination system. So you have these anthers which have little pores at the time. I should have said that when I showed the picture of all of those different Solanums, which look kind of different but more or less the same. They're all basically have little pores at the tips of the anthers, and what the bee does, and it's only female bees that can do this. Male bees can't do this. They grab the anthers with their front feet, and then they vibrate their indirect flight muscles and set up a resonance. And then the pollen squirts out onto their venters, and then their little legs can't reach it, so then there's a thing left, and they go into the next plant. So it's a very cool pollination mechanism. And one of the things I've always wanted to do, but have never done, have never ever done this, there's 1,000, I mean, I need to have several lives to be able to do all the things that I've always wanted to do, is to look at the sizes of flowers and the sizes and diversity of bees and the diversity of Solanum. And the place where we can do this is Brazil, because we have a really good understanding of what Solanums there are in Brazil. I mean, there's loads of new ones in Brazil. There's a ton of new ones. But we also have a really good understanding of the bees because of the digitization that's gone on through CRIA and SpeciesLink in Brazil. So that's something that we would like to look at. But one of the things that I found out in the Amazon this time, which I've always seen large bees buzz large Solanum flowers and small bees buzz small Solanum flowers. But actually, when I was in Manaus, I found a tiny little euglossine bee, little tiny bee, buzzing a huge Solanum flower. So it's not necessarily related to the flower size. It's, you know, what attracts the bees to those flowers? It's the nitrogen. I mean, the anthers all look yellow, but the pollen isn't yellow. The pollen's white. So these Mario, my colleague Mario at Uppsala works a lot on the physics of buzz pollination and looks at buzz pollination in some of these really interesting Solanums with different shaped anthers, because there's a lot of variation in anther shape. So anther shape and size has a lot to do with different groups. It's one of those characters that's really interesting. But yeah, they're all buzz pollinated, and they all just kind of do that. Yeah, there's one there, and then there's one here.
Thank you. You mentioned briefly about the diploid potato, and I was wondering if you could touch on that again and what
Okay,
the
so
importance of it
potatoes.
is.
Potatoes are amazing. Potatoes are amazing. So all the potatoes you see in the supermarkets are all a single species. So they're all the same species. It's Solanum tuberosum. Solanum tuberosum is an allotetraploid, which means it is the combination of two different genomes. So it's an allotetraploid, which means it has double the number of chromosomes. And because they come from two different parental genomes, that means that traditional plant breeding is very, very difficult in potatoes. So making crosses where you get things that grow true from seed is really, really hard in potatoes. And also the other reason is because potatoes are super easy to vegetatively reproduce. You just chop them up and put them in the ground, and that's all great. And that's good, but it's also really bad because it can cause problems like the Irish potato famine, which all of the potatoes grown in Ireland were a single genome. They were a single plant. And so once a new disease comes in, it goes through the whole population and kills them all, which is what happened with Phytophthora. So having a diploid potato that you can actually do traditional plant breeding on, or genetically modify in a way that's easier than with tetraploids, is what my colleague Sangmeng Wang in the Chinese Agricultural Academy of Sciences is aiming to do, to make potato breeding easier, to create potatoes, to make potatoes that will withstand climate change, or will be able to grow in more difficult places. And that's kind of one of the things that plant breeders are really trying to do, is to anticipate. And it's really hard, because plant breeding is, we've been genetically modifying plants forever. I mean, human beings have been genetically modifying plants for ages and ages. So creating something that, for conditions that you don't know there will be. I mean, we hypothesize that things are going to get drier. We don't know. You know, maybe we should be looking at those Australian solanums, which were the real winners in an earlier drying episode. So yeah, the diploid potato is about breeding better potatoes more easily without having to deal with this tetraploid genome, which is really big and really, really complicated.
There's an online question that's related, which is, what can you tell us about the wild ancestor and the domestication of potato?
Well, so it's an autotetraploid, which means that two species came together. Well, it's an allo-autotetraploid. The actual origin of potatoes is quite difficult, but we know that it happened around Lake Titicaca about 6,000 years ago. So it's relatively recent, the cultivated potato. There are lots and lots of, there are 104 wild species of potatoes, all of which have tubers. The tubers are not very nice. They're quite bitter. They have a lot of salacidine, which is a steroidal alkaloid in them. So what I find interesting is that in talking with potato taxonomists, when I very first came to Britain, I said, well, it's obvious that these people in the Andes did plant breeding. It's obvious they did this because they've selected for this thing that we've, and they said, oh, no, they wouldn't have understood about plant breeding. Really? Honestly, these are the people who lived with these plants. So they're an Inca crop. They're one of the few crops that were not banned by the conquistadores when the Spanish took over the Peruvian Inca Empire. Is that potatoes, because they were completely, life was impossible without potatoes. They were mysterious to Europeans when they first showed up. The first descriptions of potatoes are kind of about a truffle-like object, and kind of these crazed descriptions of potatoes are really fun. But yeah, so that's, I mean, I'm not a potato taxonomist. I should be. Yeah, there's a question there.
Fantastic, thank you. You made a sort of tantalizing allusion to Solanacea and witchcraft, and I wondered if you would just want to expand on anything about maybe the cultural history of this group of plants outside of cultivated varieties.
Well, what's interesting is that there's lots and lots of cultural uses of, Solanaceae, the family, can kind of be divided into two sets of different alkaloids that happen in the family. So things like mandrakes and nightshades, deadly nightshades, and datura and stuff like that all have what are called tropane alkaloids, which are 5-member rings with a little nitrogen on one of the house corners. And those are like nicotine and things. They're psychotropic. They interfere with the cholinesterase kind of receptors in your brain, and they cause the sensations of flying. So the other half of the family, of which Solanum is part, have these steroidal alkaloids, which are long chain 6-membered alkaloids, and those are very similar to the kinds of things that are put together that are used to make birth control pills. So they're these estrogen type steroidal alkaloids. So I knew a guy in Ecuador in the 1980s who was growing this Solanum, which was actually from Ethiopia and had come to Ecuador in Persian carpets that the conquistadores had brought, and they would wash them out in the streams, and the seeds from this thing, which was used as soap in Iran, would then go, and they're all on the streams in the Andes. So he was growing these plants and then trying to extract these steroidal alkaloids out of the fruits in order to create a new source for diosgenin, which is the major component of birth control pills. So So that's those alkaloids. But the psychotropic ones are these daturas and mandrakes, and they're these cholinesterase things that create this sensation of flying. And they're used as magical medicinal plants in Australia. They're used as magical medicinal plants in the Andes and in the Amazon. Ayahuasca, one of the major components of ayahuasca is Bromfelsia, which the commonest one is Banisteriopsis, which is Malpighiaceae. But Bromfelsia, which is a Solanaceae genus, is a major component of ayahuasca. But where we know the most about, well, where the culture of Solanaceae as in witchcraft actually is in Europe. So there's all kinds of really interesting early descriptions of the use of solanaceous plants in unguents and ointments that people would put on their skin. Then it's absorbed through the skin, and then it's creating this sensation of flying. So people think that may be partly where the kind of idea of witches flying around on broomsticks came from. So there's some really interesting, very early kind of descriptions of all of this. And then the whole idea of mandrakes, the whole Harry Potter thing of mandrakes, and you have to put earplugs in because they scream when you pull them out of the ground, is they, in a herbal, which was written in 1597, Gerard's herbal, which is one of the first herbals that was written in English. Interestingly, he's holding a potato flower, which is very exciting, in 1597. But he writes about the mandrake, saying that you should not believe these tales of mandrakes screaming because there's a mandrake cartel, and they're trying to keep people from digging them up because they're used there. And he said it's very efficacious if you want to cut someone's leg off, that you can give them a draft of mandrake and they won't notice anything. So yeah, so there's a lot of, and there's a whole set of plants called Brugmansia, which actually are unknown out of cultivation. So these are plants which they're not known as wild plants. They're basically all in cultivation. They're escaped now in lots of places. They're the daturas with the hanging flowers. And there's a whole, in Sibundoy in Colombia, there's a whole set of different cultivars of these which have varying concentrations of different alkaloids which are used in shamanistic practices. So there's lots of stuff like that in Solanaceae. So they're quite cool. So they're kind of a thing about, half of them are kind of magic drugs and half of them are foods that we eat all the time. So what a family to study, right?
Well, I think that's a perfect note to wrap up on. And we've got drinks at the back to do. Feel free to linger and have an informal conversation there. Thank you once again, Sandy, for absolutely fantastic.